Hey guys, today we're looking at section 11.3, which is called regulating the cell cycle. So we're going to look at here specifically exactly how checkpoints in G1 and G2 function to allow a cell to know whether or not it needs to go into either synthesis or mitosis. And we're going to talk about the proteins and hormones that are especially responsible for that type of regulation. And then also we're going to look at exactly what happens when regulation kind of goes awry and things don't go as planned. Okay. Learning objectives, we've got just two of them. So looking at that regulatory process, exactly how those proteins are able to control the cell cycle. And then we're going to look at what happens when things go wrong, like I just mentioned, which is really what we all know and can appreciate, which is cancer. Not that we appreciate it, but that we have, a basic understanding of that process. So in looking at regulation here first, there are a couple different types of regulation that we can see with cells in terms of, controlling the cell cycle and determining whether or not a cell moves from one stage into the next. So external regulators are one factor that can control a cell cycle. There's also going to be internal regulators, and I'll talk about that on the next slide. But if we start with the external, remember external means outside, so we're looking at proteins and signals that are going to be happening and occurring outside of a cell and causing a cell to do something as a result. So they trigger a response inside the cell once they're recognized by that cell. So first type of external regulator is a chemical signal. And by the way, you do want to get everything that's on this slide. These are all important items here. So chemical signals respond to events that are outside of the cell, and these include things like growth factors. So if they're chemical, that means that they are made up of molecules, right? And so molecules in this case can be growth factors, which are protein hormones that can help to stimulate things like wound healing, embryonic development, and general growth in an organism. You've probably heard of human growth hormone. That is an example of a growth factor. So when we use the word growth factor, we're talking about growth. And with growth comes more frequent cell division, right? Remember we talked about in 11.1, cells can only grow so large. So if we're looking at growth of an entire organism or an organ or a tissue, growth factors are going to help to stimulate cell division, not necessarily larger sizes, but increased frequency of cell division. Okay, so those are coming from outside of the cell. The second type of external regulator is a physical signal, and these are more of those things that we would be able to identify using our senses. So if we think of the cell as like our human body, as if we have senses within a cell, they can sense what's around them based on touch, based on temperature, based on their general size. And so we know that for normal cells, and you can see here in this image, at the bottom, this is what happens with eukaryotic cells that are grown in a petri dish in a lab under normal conditions. If they're functioning properly, all those cells will grow until they fill up that space, but they don't grow on top of each other. Once they start bumping into each other, they're going to stop. Okay, and at that point, when they start bumping into each other, either they're going to initiate cell division or they're just going to simply stop growing and continue doing their normal functions. All right, but that touch, the pressure that they feel on the exterior is going to be a signal to the cell that either it needs to divide or it needs to stop growing or something of the sort. Okay, so that's an example of, again, an external regulatory factor. It's something that tells a cell whether or not it needs to go through this process of cell division. Okay, so if we compare that then to internal regulators, these are things that we see inside of a cell, right? If it's internal, it's on the inside. And internal regulators are going to be something that also stays within a cell. So they're produced in a cell and then they stay in that same space. In general, things that are happening on the outside are going to help to trigger things on the inside, right? So the growth factors we looked at, those chemical signals, and then also the physical signals, the way that those signals are translated to a cell is with the help of these internal regulators. So internal regulators are almost always proteins. The two most important protein regulators that are going to be active inside of a cell are called kinases and cyclins, all right? And kinases are actually more appropriately called cyclin-dependent kinases, but here we just have the word kinases. These are both proteins that help to kind of function like stoplights in the cell cycle. So they're going to be what actually tells a cell to stop as a result of something happening on the outside. They would be what tells a cell to continue moving through the pathway. They may be proteins that are created if there are errors in things like DNA replication, if organelles aren't copied properly, if the cell hasn't grown enough, and so on. So these are going to be kind of the most direct influencers in terms of really making sure a cell is doing the right thing and only dividing when it's most needed. Okay. So when we talk about cell division, one of the things that gets added in here is this process of apoptosis. Apoptosis is a normal feature. It's what we find in all healthy organisms. You have gone through apoptosis in some tissues of your body, as have I and every other human that's, in our class or in this building. And I know that because we all lack things like webbed fingers and hopefully also webbed toes. But what we see in apoptosis, this is really defined as programmed cell death. And it sounds kind of like, ooh, doom and gloom. But when we look at programmed cell death, this is actually good things. So apoptosis can be used to either, one, get rid of cells that could be potentially harmful, where maybe there are things that are going wrong that can't be fixed as part of the cell cycle. It may even be that we have errors and malfunctioning cyclins and kinases, which could really be a problem in a cell because then there's that whole lack of order in the cell cycle. But more often than not, apoptosis happens simply in cells that are just not needed anymore. Okay, so what we're looking at in this picture, we have a developing fetus here, which is one stage of development when a baby is being produced and is growing in a mother's womb. You can see lots of different features. There's the skull and there's an eye and there's some little arm buds and hands and there's legs down here with probably some little feet and the spinal cord running along the back. And what they're trying to show you kind of side by side is how different this looks compared to this. And obviously, yes, it looks more alien-like here, but they're pointing at one specific feature, which is the fingers. All right. And so what we're looking at here specifically, when babies are developing, we know that they all have webbing between their fingers and their toes. And so what we know happens with that, we understand that it's there for protection. A lot of times when I ask the question about this in class, I get kids that ask me if it's because the baby needs to swim when in the womb. And no, that's not the case because there's just simply not that much room in there. But the webbing itself helps to protect and allows these bones to form so that they're nice and straight and not like broken and backwards and twisted around when the baby is first born. However, around the time that the baby is supposed to be born and development is kind of reaching its peak and is almost done, that webbing is going to go away, and it goes away through apoptosis. And again, it's because it's simply not needed anymore. Okay. We see this with lots of tissues that are forming and then breaking down as part of infant development. And again, it's just a normal process. And really, for adults, it now becomes kind of a protected thing, because if there is something that goes wrong, we can still get rid of and terminate cells that are no longer needed, okay, or that could potentially cause harm. So the way that that's done, you can see here in this bullet I've got, it's caused by the cell's production of self-destructive enzymes or proteins. And these are really going to come out of lysosomes. If you remember, the job of a lysosome is to help break down things that are no longer needed by the cells. So essentially in apoptosis, those lysosomes are triggered to burst open, and all those enzymes that they contain within them are going to start breaking down the actual cell. Okay. Sounds painful, not painful. And again, also super helpful. All right. So what can happen though, if apoptosis doesn't occur like it should, if we lack regulation in our cell cycle, we know that one of the things that can happen as a result, and in turn, is going to be cancer. Cancer is a topic that we all know some things about. I would assume we've all probably known someone who's had cancer or has cancer, and that's just something that's become pretty typical in our population, in our species. But lots of different organisms can develop cancer. Cancer in general is just a term to represent uncontrolled cell division, and that's it. So by and large, by itself, to me anyway, if I think of uncontrolled cell division, well, that doesn't seem all that bad, right? It's just cells that are just dividing without that regulation. But the problem is when cancerous cells develop through this uncontrollable cell division process, we're talking about cells that are no longer doing what they're supposed to be doing. And so what that means is that if we have something like cancer that's developing in the liver, let's say, those cancerous liver cells are no longer going to function like normal liver cells. So not only do they divide rapidly and uncontrollably and kind of disorganized, they're going to also completely lack the function of the tissue in which they're found. And so over time, because they divide more rapidly than your normal functional liver cells, they can kind of overtake the organ, develop into tumors that basically can shut down the entire organ over a period of time. And that's where you run into more serious issues with this, right? Simple uncontrolled cell division by itself doesn't imply that there's that much harm being done, but it's the effects of that, right? It's the fact that over time you have more and more cells of this tissue that are not doing their job, which means the tissue in the organ can't function. Okay. And if that occurs in enough places in the body, that can shut down an entire organism, right? So we know cancerous cells form disorganized clumps that we call tumors. These are those lumps and bumps that you probably heard of. There are two different types of tumors. We have benign, and that's how you say this word right here, benign tumors. They remain clustered or localized, which means they all stay in one place. A lot of times, benign tumors are not super harmful or dangerous, and a lot of times they can just simply be removed. And if that's the case and nothing has spread before they were removed, you tend to kind of get it all, and the individual lives for a much longer period of time. Now, on the other hand, malignant tumors are what represent what we call metastatic cancer because they metastasize right here. And metastasize is a fancy scientific word to represent things that are breaking away or spreading. So malignant tumors are those types of cancers that spread throughout the body. And it can just be spreading to one place or multiple. But the key is malignant tumors have to use the bloodstream to spread from one place to the other. So these are types of cancers that have kind of hopped into the bloodstream. Those cancerous cells have enlarged in their tumor large enough so that they can actually kind of interact with the bloodstream, and they kind of use those blood vessels to travel all over the body. And that's really where you run into some serious risk and danger. Okay. So what tends to happen in these cancerous formations, so where we have, going from benign to maybe a malignant tumor, we have just, all this is going to start with just one cell. And one cell begins to divide abnormally. And as a result of that, abnormally also implies that it's going to divide more frequently than it would normally. And so it's going to kind of very quickly start to grow. And sometimes these things can spread more quickly than we would have ever thought. They produce a tumor. Those tumor cells start to displace normal cells and tissues, meaning, again, like in this example, these normal cells are going to be pushed out of the way, so much so that usually what ends up happening is the whole tissue ends up shutting down. If that tumor grows large enough and, again, gets into the bloodstream like we're seeing right here, it can go on and spread to other parts of the body. Okay, but cancerous cells are truly just, and I can't emphasize this enough, cancerous cells are truly just our cells. Like, it's not a virus. It's not something that's specifically trying to cause you harm. It's cells that are not functioning properly. Something's gone wrong, right? And they're not following the directions that they should be following. So in all cancers, again, what they all share in common is this uncontrolled cell division. So we don't have the regulation that we normally see and that we want to see in normally functioning cells. Those internal and external regulators aren't having the impact and the control that they need to. So usually when that happens, that can come from dysfunctional genes. And those dysfunctional genes can either be inherited as mutations. For instance, we have genetic tests for the BRCA genes, which we know are associated with the development of breast cancer. There are some cancers that we know are linked to genes specifically, but then there are others that we know are more environmental, right? Skin cancer is environmental. Lung cancer tends to be more environmental. There aren't specific genes associated with those. And so it can have a genetic kind of basis, but it can also be a lifestyle kind of thing. Or, I mean, truly, some of these things, there is no rhyme or reason. It's just something goes wrong at one point and the cell doesn't recognize it and it just multiplies, right? So sometimes they're more unpredictable. What they all share in common, though, is the lack of regulation, again, by specifically kinases and cyclins. So usually there's something wrong with either the kinases or the cyclins, again, are internal regulators that function as those stoplights in the cell cycle. So if those stoplights aren't functional, obviously you can think about on a road if you want to compare it that way. If my stoplights aren't functional and if drivers aren't paying attention, it would become really easy to kind of smash into each other in an intersection. And that's what happens in a cell. If those stoplights aren't functioning properly and if the cell's not paying close enough attention, it just runs out of control. Okay. So cancerous cells, again, like I mentioned, do not carry out normal functions. They grow uncontrollably. They can crowd out cells that are functioning properly. And in this picture here, you can see how this compares to what we saw earlier, in that we are having this tumor kind of forming here. There's lumps, there's bumps, there's things, cells growing on top of each other, and so on. And that's exactly what you see in a lab if you're working with and growing cancerous cells. So in looking at cancer specifically, there are a few treatments that we know are widely used. Some are more successful for some individuals, but truly cancer differs for each individual. And so treatment can be kind of variable, right? I mean, we can use different combinations of these. So the three major options that we have in terms of cancer treatments are surgery if we have tumors that are benign, right? If you have tumors that are localized in one area, surgery can usually be used to remove those cancerous tumors. And along with that, they often also remove lymph nodes to see if the cancer has been spreading at all. Radiation can be used also to get rid of cancerous cells. And the way that works, we're using wavelengths of light essentially to damage DNA so that it no longer can allow a cell to replicate. Or if a cell replicates, it's going to lack the DNA codes that it needs to be able to function. And so with radiation, this is, again, for more of those kind of localized tumors. You can't use radiation on the whole body because the effect of that would be to essentially kill off every cell in the body, and we can't have that. So we use radiation in specific areas to target cancerous cells, all right? The one thing on this list that is kind of involving the entire body is chemotherapy. Chemotherapy is the use of drugs and medicines to try and remedy and rid the body of cancerous cells. But this is always targeting strictly rapidly dividing cells. And so while it will kill off cancer cells, the goal here is going to be that it doesn't kill off too many healthy cells. And as we've talked about in this chapter, there are lots of cells that divide fairly rapidly, but are actually supposed to be dividing fairly rapidly. And chemotherapy can't distinguish between those and cancerous cells. So chemotherapy is exactly the reason why cancer patients who are undergoing this treatment may lose their hair, because hair follicles, as we've mentioned, are rapidly dividing cells. They may have more issues with their skin, right, like eczema or rashes or bruising, because rapidly dividing cells exist on the skin, right? They can have issues with nausea and digestive upset because our stomach lining divides very frequently also. And so you're killing off all of those cells, which makes it really difficult to consume and digest food, right? So anything that's dividing rapidly is targeted by chemotherapy. And even though it can address kind of the whole body, there's a balance here between, okay, how often should we be using this to get rid of the cancer cells, but also then not kill too many of the normally functioning and important cells in the body. Okay, that's where it becomes kind of tricky to design or develop those treatments. This lovely little picture right here is of a mole on someone's skin, and this is my normal time when I have a little PSA about skin care, right? And also visiting a dermatologist if you notice anything kind of odd. This mole is very odd. If you ever see one of these like this on your body, you definitely need to go see your doctor and have that checked out. There are always some things that you're supposed to look at with your moles, and one of those is color. And notice this color is different. So for any normal mole, you should see the same color kind of throughout. You can see different colors here. You can see I've got like a glare because it looks like it's raised. It's not just one kind of flat surface. There's like bumps and lumps here. It is not symmetrical, which is also an indicator to a dermatologist that this is not a normal shape. And I can't remember what the other one was, but those are kind of the general items that you need to look for when you look at things like moles on your body. And it tends to be, you know, as you get older, like me, you tend to develop more of these things, not like this, hopefully, but more moles as you are exposed to more and more sun in your lifetime. And so this is something we all need to kind of watch out for and be aware of. All right. All right, so that's all I've got for section 11.3. As always, email if you have any questions. We'll go over this in class as usual or on Zoom, and I'll talk to you later. All right, bye, y'all.